A very good morning to all of you. Today we are discussing Mendel's law of inheritance. Yes, we can discuss monohybrid cross and dihybrid cross. Mendel selected garden pea for his experiment. Do you know why did he do that? Yes, there are some reasons are there to select garden pea for his experiment. They are many varieties were available in alternative forms of a character. That is one of the reasons. Then these varieties were available in pure forms. That read true. We know that these pea plants are self-pollinated. Normally they are self-pollinated, but that can be prevented by removing corresponding reproductive parts. That means we can remove the anther of the flower and we can cross-pollinate artificially. Then the next point, the lifespan of the pea plant is short. The lifespan of the pea plant is very short and many generations can be obtained and studied in less time. These are the reasons why the Mendel selected garden pea for his experiment. Mendel selected 7 main characters for his experiment in garden pea. They are flower color, seed shape, seed color, pot shape, plant height, flower position. These are the 7 characters he selected in garden pea. Then each character has 2 different traits. One is dominant, another one is recessive. Yes, in flower color, 2 traits are there, purple and white. Purple is dominant, white is recessive. Like that, seed shape round and wrinkled, seed color yellow and green, pot color green and yellow, pot shape inflated, constricted, plant height tall, short, flower position axial and terminal. These are the 2 different traits of each characters. Mendel crossed pure breeding varieties, first by taking only one feature at a time, that is called monohybrid cross. For example, true breeding yellow pot crossed with true breeding green pot. Here, he took only one character at a time. Pot color is a character. That character he took. That's why that is a monohybrid cross. Only one character. Then this pot color has 2 traits, green and yellow. Then he took true breeding or pure breeding varieties. What is the meaning of that? Because both the parents are homozygous. One parent is homozygous recessive, another parent is homozygous dominant. But one character he took, that's why that is monohybrid cross. But in the case of dihybrid cross, 2 features he took at a time. He took 2 features at a time, that is called dihybrid cross. For example, seed shape and seed color, these 2 are 2 characters. For each parent he took 2 characters at a time. Then seed shape and seed color he took. Then round and yellow, these 2 are dominant traits. Wrinkled and green, these are recessive traits. Then both are pure breeding varieties, because you know that what is pure breeding or true breeding. Alleles are homozygous, but 2 characters are there. Then that's why that is dihybrid cross. Phylal generation. The first, second and third generations of the offspring are known as first filial, second filial, and third filial generation and are denoted by the symbol F1, F2, and F3 respectively. Here you can see parental generation. Both the parents are pure breeding. What is the meaning of that? Both the parents are homozygous. One is dominant, another one is recessive. But both are homozygous. Then what will be the first generation? The first filial generation, all the progenies are dominant. That shows purple flowers. But that is heterozygous. Then the second generation is F1 self. Then we will get second generation. That is second filial generation. Let us learn monohybrid cross. Mendel crossed pure breeding varieties. Mendel selected one feature at a time. That's why that is called monohybrid cross. We can go through one example. Here, axial flowered plant crossed with terminal flowered plant. Both are pure breeding, homozygous. Then here, which is the character Mendel selected here? The position of the flower. Then two varieties of plants we are getting from this, axial flowered and terminal flowered plants. This axial flowered plant is which type of gamete we are getting? Only capital A. Then terminal flowered plant we are getting small letter a. These two gametes fuse to form axial flowered is plant. In F1 generation, all the progenies from F1 generation are axial flowered plant, but they are heterozygous dominant. After F1 selfing is F2 progenies we will get. Then the parents of F2 progeny, they are heterozygous dominant. Then they are heterozygous, that's why from each parent we will get two types of gametes. Here you can see two different alleles are there in each parent. That's why from this parent we will get capital A and small letter a as a gamete. Yes, from this parent we will get capital A and small letter a again. Then the progenies. What are the possible progenies we are getting from these parents? F1 selfing, after F1 selfing. Here you can see is capital A gamete from one parent fused with capital A gamete from another parent. Then we will get axial flowered plant. Capital A gamete from one parent fused with small letter a from another parent. We will get axial flowered plant. But here this is heterozygous. Here this is homozygous. Right? Then here again small letter a gamete from one parent fused with capital A gamete from another parent. Then we will get axial flowered plant. This is also heterozygous. Then small letter a and small letter a fused to form is terminal flower plant. Then we can go through the ratios. Then in F2 progeny, 3 axial flowered plants we are getting, then 1 terminal flowered plant. Then the phenotypic ratio, what is phenotype? The expressing characters, 3 axial flowered plant, then 3:1 phenotypic ratio of monohybrid cross, that is 3:1. Then genotypic ratio, what is the genotypic ratio? Here you can see this is homozygous dominant, this is heterozygous dominant. These two are heterozygous dominant. Then 1 homozygous recessive. Then genotypic ratio, genetic constitution, that is 1:2:1. This is the genotypic ratio of this F2 progenies or F2 generation. Here, when it will be possible, phenotypic ratio is 3:1. Then genotypic ratio is 1:2:1. In all the cases, it will not be possible. Both the parents are heterozygous. Then only this ratio will be possible. Both the parents are heterozygous. Monohybrid. Then the monohybrid ratio of or monohybrid phenotypic ratio is 3:1. Monohybrid genotypic ratio is 1:2:1. Monohybrid phenotypic ratio is 3:1. Monohybrid genotypic ratio is 1:2:1. When is it obtained? When both the parents are heterozygous dominant, then only we will get is phenotypic ratio 3:1 and genotypic ratio is 1:2:1 in monohybrid cross. The kind of ratio obtained by crossing for two different traits of a single character is known as monohybrid ratio. We can go through one example. Is in these two varieties of plants, tall and dwarf, which is the character, single character we took, that is the height of the plant, that is the character. Then that is character has two varieties of plants, is tall and dwarf. Pure tall plant crossed with pure dwarf plant. Then the possible gametes we are getting from the tall plant, that is capital T. The possible gametes we are getting from the dwarf plant, that is small letter t. And these two gametes fuse to form is heterozygous dominant plant, that is all the F1 progenies will be, this is F1 progeny, yes, these F1 progenies will be heterozygous dominant. All the progenies will be tall. After selfing this F1 generation, then we will get the phenotypic ratio will be 3:1, genotypic ratio will be 1:2:1. Why? These parents will be, after selfing, F1 selfing, the parents will be heterozygous dominant. Then two different types of gametes we are getting from each parent. Is from this parent, that is possible gametes are capital T and small t. Here also the other parent, the gametes will be capital T and small t. Then two possible gametes are getting from each parent. Then in this case, the genotypic ratio will be 1:2:1. Then phenotypic ratio will be 3:1. Here you can see is 3 tall plants and 1 dwarf plant. Then phenotypic ratio will be 3:1. Then 1 homozygous dominant plant, 2 heterozygous dominant plant and 1 homozygous recessive plant. That's why genotypic ratio will be 1:2:1. Here I am giving another type of crossing without using Punnett square. Yes, the same example I am giving, pure tall plant crossed with pure dwarf plant. Then from the pure tall plant we will get only one type of gametes, capital T. Then from pure dwarf plant we will get only one type of gamete, that is small letter t. Then these two gametes fuse to form capital T, small letter t, that is the progeny, possible progeny, that is heterozygous dominant progeny. This progeny we will get in F1 generation. This is F1 generation. Then all the plants will be tall, but that is heterozygous. This heterozygous progeny, after selfing, we will get two types of gametes, capital T and small letter t, from each parent. We will get two types of gametes from each parent. Then the crossing will be like this, as without using Punnett square. Then here you can see capital T gamete from one parent crossed with is the capital T gamete of another parent. Then the progeny will be tall. Then again capital T gamete crossed with small letter t of another parent, then that is tall but heterozygous. Then here again small letter t from first parent crossed with capital T of another parent, then the progenies will be heterozygous tall. Then here you can see small letter t of one parent crossed with small letter t of another parent, then we will get dwarf plant. These are gametes. Capital T is one gamete, small letter t is another gamete. Gametes we are representing like this. In this way also we will get is phenotypic ratio 3:1, then genotypic ratio 1:2:1. But the method of crossing is different. In this way you can cross without using Punnett square. Here I am showing different types of parents and its F1 generation. The first case, that is monohybrid cross, case 1, tall plant cross with dwarf plant. Then F1 generation, all the plants will be tall, heterozygous tall. Here you can see. Then in case 2, the parents will be heterozygous. Can you say in this case what is the genotypic and phenotypic ratio? When the parents are heterozygous dominant, then phenotypic ratio will be 3:1. Then genotypic ratio is 1:2:1. Right? This we already mentioned. Then the case of case 3, heterozygous dominant tall plant crossed with homozygous recessive dwarf plant. Then in this case, what are the possible gametes? Question will come like this. The possible gametes from the first parent, that is capital T and small letter t, but possible gametes from the second parent, that is only small letter t. Both the gametes will be small letter t. Then what are the possible progenies in this case? That also very important question. Here you can see is tall plants. 2 tall plants, 2 heterozygous tall plants we will get. Then 2 homozygous dwarf plant we will get. You can make Punnett square and close it, and you can find the progenies like this. This is very important. Then based on the question you have to answer what they are given. If they are asking is cross with heterozygous dominant parent with homozygous recessive parent, then the progenies will be like this. Then both the parents will be heterozygous, then the progenies will be like this. Then one parent is pure tall, other parent is pure dwarf, then all the progenies will be heterozygous tall, dominant tall. Dihybrid cross. 2 characters or features are taken together for crossing. That's why that is called dihybrid cross. Here you can see one example is 2 parents, both are having 2 characters. 2 characters for each parent. That's why that is dihybrid cross. What are the 2 characters we are taking here? Color and shape of the seed. Color and shape of the seed. But 2 is varieties are there. 2 pure breeding varieties are there. One is homozygous dominant, another one is homozygous recessive. One parent is round yellow. Parent produces round yellow seed. Another parent produces wrinkled green seed. Then 2 characters we are taking for each parent. Then what are the possible gametes we are getting from each parent? We are getting from first parent, homozygous dominant parent, capital Y, capital R, homozygous recessive parent, small y, small r. Here, while representing gametes, we have to include 2 characters. Here, capital Y, capital R, we have to include in that gamete, but in a monohybrid cross, we are writing only one letter there, but here 2 different letters you have to represent because 2 characters are there. In this way, you have to represent the gametes. Then, after fusion of these 2 gametes, F1 progenies will be yellow round. Heterozygous dominant character is there in F1 progeny. Then all the F1 progenies will be yellow round. But these 2 characters will not mix up. They are independent. While writing, you have to represent like this. Then after selfing, then we will get F2 generation. Then what are the possible gametes we are getting from heterozygous dominant parent? Heterozygous dominant parent, we are getting 4 different types of gametes. These are the possible gametes. You can see here capital Y, capital R we can take. Remember, while writing is gametes, 2 characters will be there in each gamete. Right? Because that is dihybrid cross. Then capital Y, capital R we can take. Capital Y, small r we can take. Right? Then small y, capital R, small y, small r. Then these are the possible gametes we are getting from heterozygous dominant parent. Then in F2 generation, the phenotypic ratio will be 9:3:3:1. Remember, when this phenotypic ratio will be 9:3:3:1 in dihybrid cross, the parents will be heterozygous dominant. Very important, parents will be heterozygous dominant. Then only in dihybrid cross, phenotypic ratio will be 9:3:3:1. Then genotypic ratio of dihybrid cross is very complex. Yes, that you don't want to learn in this 10th. In dihybrid cross, phenotypic ratio will be 9:3:3:1 when both the parents are heterozygous dominant. This kind of ratio obtained by crossing for 2 different traits of 2 characters is known as the dihybrid ratio. This dihybrid ratio is 9:3:3:1 when both the parents are heterozygous dominant. This is very easy to remember. I am giving the same example. Once again, we can explain here. Seed shape and seed color we are taking here. That's why that is dihybrid cross. 2 characters we are taking. Seed shape, 2 traits are there, round and wrinkled. Round is for dominant character. Wrinkled is a recessive character. Yes, capital R we are taking for round because that is dominant. Small r for wrinkled, that is recessive. Then another character, that is seed color. 2 traits are there, yellow and green. Yellow is dominant over green. Then that's why capital Y for yellow, small y for green. In parental generation, round yellow crossed with wrinkled green plant. Round yellow plant crossed with wrinkled green plant. Then all the progenies in F1 generation will be round yellow. All the progenies are producing round yellow seed in F1 generation. After selfing, these F1 selfing, F2 progenies, 9 is progenies are producing round yellow seed. 3 progenies are producing wrinkled yellow seed. 3 progenies are producing round green seed. 1 progeny is producing wrinkled green seed. This is the ratio. You know that each is progeny or each plant will produce is so many seeds. Then 9 plants will produce round yellow seeds. Then just imagine how many seeds are there in each plant, right? Then this is only the ratios. Then 3 plants will produce wrinkled yellow seed, 3 plants will produce round green seed, then 1 plant will produce wrinkled green seed. Then this is the F2 generation. Then dihybrid phenotypic ratio will be 9:3:3:1 when both the parents are heterozygous dominant. Some generalized principles based on the breeding experiments of Mendel. Each pair of contrasting characters depends on a pair of genes. Each individual carries such genes in duplicate. An individual produces gametes that have only one member of pair of genes. Here you can see each pair of contrasting characters depends on a pair of genes. Each individual carries such genes in duplicate. Here you can see. Yes, these genes will be in duplicate. Each individual carry a particular type of genes in duplicate. An individual produces gametes that have only one member of pair of genes. That produces only one member of pair of genes. Fertilization of gametes restores the duplicate condition of genes. Then in gametes, only one member of pair of genes, but after fertilization of gametes from different parents, yes, that will restore. The duplicate condition of genes will restore after fertilization. Sex cells with respect to the genes they contain get fertilized at random. You know that is Y chromosomes you can see only in males. Some Y-linked genes are there, that genes you can see only in males. Then sex cells with respect to the genes they contain get fertilized at random. Mendel's law of inheritance. Mainly there are 3 laws are there. Law of dominance, law of segregation, law of independent assortment. First we can learn law of dominance. Out of a pair of contrasting characters present together, only one can express itself while the other remain suppressed. Here you can see one example. Pure tall plant crossed with pure dwarf plant. Then in F1 generation, all the progenies are tall. Why all the progenies are tall? Yes, this shows the dominant character will express, the recessive character will suppress. Here, if you are checking in F1 generation, all the progenies are tall, but they are heterozygous. If you are seeing the genetic constitution, genotype, heterozygous dominant. Yes, how we can find out that is a dominant character? Because which character is expressing? Which character is not seen here? Then we can say the seen character, that is dominant character, then suppressed character, that is recessive character. Then out of a pair of contrasting characters present together, that you can see in heterozygous condition, right? Yes, only one can express. Capital T is there. Then that dominant character will express. The recessive character will suppress. That is the law of dominance. Law of segregation or law of purity of gametes. That is the second law. What is law of segregation? The two members of a pair of factors separate during the formation of gametes. Two members of a pair of factors separate during the formation of gametes. Here you can see is these are the pair of factors that will separate during the formation of gametes. You can see only one T here, right? These pair of factors will separate at the time of gamete formation. Here also you can see. Here is this is the parent, then you can see at the time of gamete formation, these two factors will separate. That we call it as law of segregation or law of purity of gametes. Law of independent assortment. What is the law of independent assortment? When there are two pairs of characters, the distribution of the alleles of one character into the gametes is independent of the distribution of the alleles of the other characters. Yes, this is Mendel's third law. It defined as when there are two pairs of characters. Here you can see two pairs of characters, then that is applicable for dihybrid cross. Then the distribution of the alleles of one character into the gametes. Here you can see at the time of gamete formation, the distribution of the alleles of one character into the gametes is independent of the distribution of the alleles of the other character. Then these two alleles from different characters are independent to each other. That is the law of independent assortment. This you can see only in dihybrid cross, because here you know that in the definition two pairs of characters, then that will not applicable for monohybrid cross. This is very important. Applications of Mendel's laws. A knowledge of the basic Mendelian principles give us an idea about new combinations. Very important. Idea about new combinations in the progeny of hybrids and enable us to predict their frequency. The next point, such information is of great importance to both plant and animal breeders for producing better breeds. This is very helpful for plant and animal breeders to get better breeds. New types of plants with new combinations of useful characters can be produced by hybridization. Then through this law is new combinations we can try. These are the applications of Mendel's law. Mutation. What is the definition of mutation? Mutation is a sudden change, very important, sudden change in one or more genes or in the number or the structure of chromosomes. It is called mutation. You have to remember this term, sudden change in one or more genes or in the number or the structure of chromosomes. The mutation alters the hereditary material of an organism's cells and results in certain characters or traits. And different characters or different traits will develop due to mutation because it alters the hereditary material of an organism's cells. It means which is the hereditary material? Chromosome. This chromosome will alter due to mutation. Then what is the result? It results in certain characters, new characters or new traits will develop. For example, sickle cell anemia. Here you can see blood cells are sickle shape. Some blood cells are sickle shape. Yes, this is due to mutation. Then another one, radioactive radiation. Due to radioactive radiation, mutation can occur. Revision. Define the given terms. Dihybrid cross, monohybrid cross, phylyl generation, mutation, law of dominance, law of independent assortment, law of segregation. These are the important definitions coming from this chapter. If you have any doubt in monohybrid cross and dihybrid cross, you can ask me. Thank you, children. Have a good day.